Welcome to the 2021 ISV International Congress. We welcome you to our online virtual Congress this year. ISV has had a long history of providing these congresses in person. This is our 17th event, but this year will be unique and different than we've ever done before. So over the last year, we've all dealt with quite a number of trying events associated with the COVID pandemic. Last year, ISV was unable to host its Congress in person, and in lieu of that, we ran a series of online short webinars, which were quite successful for ISV. But now we're holding our annual Congress just for the next 3 days. We're glad that you've been able to join us. COVID has really changed our lives. It's made things more trying, but one of the good things that has sort of come out of the pandemic is the fact that vaccines are powerful and they can really make a difference in how we live our lives. And it's a great time to be a vaccine scientist and vaccinologist here in this world. The invention of numerous COVID vaccines over the past year is a culmination of years of research sponsored by private industry, government agencies, academic institutions that led to our ability to make COVID vaccines so rapidly and quickly and bring them to people who needed them around the world. ISV is dedicated for vaccines. We are a society that is positive for vaccines. We want to support all aspects of vaccines and vaccinology, including public health, government policy, basic research, clinical science, corporate development of vaccines. And so I think you will see that in our Congress today and over the next 2 days, how it's a wide, diverse number of people who work in this field and have different points of view that we'll bring to our Congress this year. We have numerous speakers who are going to give presentations, but there's also posters that are being presented. There are vendors that are showing their wares and you can talk with them about their technologies. There are interactive fellow talks that you'll talk to our senior fellows in the society. So please explore the entire venue. Look at all the different locations. It's really quite an interactive online system that you can take advantage of. Talk with your fellow presenters, talk with your fellow colleagues, talk with the vendors about their technologies. We also want to remind you that we have our general annual meeting this year on Tuesday afternoon, which is in the US time. All members are welcome to attend this general meeting. This is an election year, so we will be actually having presentations on the election and the process for electing new officers and board members. So please see if you can attend that. We would like to have you as a member of ISV, and if you can attend that general meeting tomorrow afternoon. So this Congress is going to be co-chaired by 3 different presenters. Today I'm going to introduce to you one of the co-chairs, Dr. Manon. She is leading this year's Congress, and I will turn over to her and let her introduce the Congress this year and our keynote speaker. Dr. Cox.
Thank you, Ted. So I also like to welcome everybody for joining us today. We're having a very exciting program. I'll speak a little bit to that, but first of all, I would like to thank my co-chairs, Dr. Guus Rimmelswaan and Laura Palomares, for helping putting this program together. Guus will be actually hosting the rest of this day, and Laura will be hosting tomorrow, and I will be hosting Wednesday. I want to thank the scientific committee that helped putting a program together at relatively short notice. They've been very helpful. Thank you for all your ideas and suggestions. And as Ted already mentioned, we have a very broad program with multiple speakers, keynote speakers, and also posters. So I really encourage everyone to browse around in the website because it's very interactive. I definitely want to thank Sean Liu and Ted Gibson, who really have done a lot of work making this administratively flow and putting everything together. So without further ado, I'd now like to introduce our very special keynote speaker of today, Dr. Ian Lipkin. And Dr. Lipkin is the director of the Center for Solutions for MF and CFS, the John Snow Professor of Epidemiology at the Columbia University Vagelos College of Physicians and Surgeons. And he is director for the Center of Infection and Immunity with the Manon School of Public Health at Columbia University. Dr. Lipkin is internationally recognized for his contribution for global public health through innovative methods he developed for infectious diseases, diagnosis, surveillance, and discovery. These advances have been critical in replacing culture-dependent methods of global health management, and we'll learn hopefully a lot about that today. Dr. Lipkin has been on the forefront of the outbreak response to many of the world's recent outbreaks, including the West Nile virus in 1999, SARS in China in 2003, MERS in Saudi Arabia from 2012 to 2016, then Zika in the United States 2016, encephalitis in India, and COVID-19, of course. Dr. Lipkin went to China in late 2020 to consult with colleagues at the CDC during the early assessment of the COVID outbreak, and he contracted COVID via community transmission in March, living in New York, and maybe he'll share some of that experience with us. And what I thought was very interesting, because everyone can read his CV in the conference environment, but Dr. Lipkin also served as a scientific advisor for the Soderbergh film Contagion that many of you may have seen. He has received many prestigious honors, and without further ado, I'd like to open the floor for you, Dr. Lipkin. We're really looking forward to your presentation. Thank you.
Thank you so much for this generous introduction. And I'm going to now switch to my slides. And what I like to say is that while my lab and my colleagues focus on trying to identify targets, you are the ones who actually do something about it. And I think that's absolutely critical, and I'm really delighted to be part of this conference. Can everyone see this hearth with various bugs on it?
Sorry.
Apparently we could.
It was the wrong one.
We could, and now the screen is gone. Good. We're good again.
This works again?
No, it's on the
Yes.
wrong screen.
It's on the wrong screen. What do
I
you
think
mean?
so, because it's, sorry. So sorry.
Dr. Lipkin, you are one of the only ones who's doing this live, so your challenge is huge today.
Well, I don't know about that. If I had some jokes, I would share those with you, but I think we'll just wait.
Everybody gets to ask you questions.
Okay. Does that work? Yes? You can see the screen with the hearth and all the bugs that are the targets for us? Great. Thanks very much.
It
So,
would be better if you were to swap your screen at the top.
Kelly, I think we did exactly the opposite of what we wanted to do. So in any event, what I'm trying to emphasize here is that although I'll be talking about viruses primarily, we study many other agents. And it's important to recognize that bacteria, fungi, other pathogens are going to be important targets as well. Now, as Manon said, I'm the John Snow Professor of Epidemiology, so I like to start these lectures with John Snow. And this was his work in 1854 in cholera, where he implicated this pump as the source of the outbreak. And last week, we had the 167th anniversary of him breaking off the pump handle. Now, for those of you who've been to London and look for this pump, you will find one precisely where it was originally located, but that's a facsimile. The actual bona fide pump is located in the London School for Hygiene and Tropical Medicine. Of course, now when we investigate infectious agents and outbreaks and other things, we use different techniques. Here's a heat map that illustrates many of the important pathogens that have been identified recently, including SARS-CoV-2. But again, emphasize that we have not only viruses, but bacteria as well. And the tools that we use primarily are sequencing. Increasingly, you'll see that there will be a shift to thinking about serology as well. And I'll come back to that in a moment. Now, I didn't initially intend to go into pathogen discovery. It really came about as a function of my second residency in neurology in San Francisco in the early 1980s, when we had these cases that began to appear in San Francisco of people with unexplained illness associated with immunosuppression. And as you all know, it took 2 years from recognition of the disease to identification of causative agent. And there was a lot of squabbling back and forth between scientists in the US and in France about who deserved credit. I think at this point, there's no question that it was the Pasteur group that made the seminal observations. But I decided that there should be ways in which one could do this much, much more rapidly. I went to work with Mike Oldscone at Scripps Clinic, who told me shortly after I arrived that there was a vaccine right around the corner for HIV. So I should shift instead to work on LCMV, which I began to do with Rafi Ahmed and others. But the thing that really struck me when I was getting back to this whole concept of trying to find agents and implicate them in disease was whether or not there was something that was high profile and intriguing that I might focus on. Rudy Rott in Giessen and Hilary Kaprowski had described antibodies directed against a virus that had not been isolated or characterized in people what was then known as manic depression, now known as bipolar disorder. So by using subtractive cloning over a period of several years, we identified the first cDNAs representing components of that agent. And I didn't have the wherewithal to do any sort of sequencing. So there was a lot of molecular gymnastics associated with proving the link in situ hybridization, hybrid rest experiments, and enormous amounts of radioactivity. And of course, now, in contrast to what we did then, which took such an enormous amount of effort, we can rapidly identify infectious agents. The first time we became involved with an outbreak was with West Nile virus. And I show this to illustrate the point that I think this was critical in really demonstrating the power of One Health, because there were two completely independent lines of investigation. One that was focused on what was occurring in animals. And this was work that was really prompted by observations by Tracy McNamara then at the Bronx Zoo, which then led to work with the USDA and to some extent with the military. And then in the New York State Department of Health and Wadsworth Center, where we began working with this, looking at ways in which we could take a differential display to try to identify that. And these two lines came together. And at that point, there was a general accounting office investigation to ask the question of why people hadn't been thinking about the importance of One Health. This was incredibly instrumental, I think, in demonstrating that we needed to begin thinking about diseases in wildlife and in domestic animals and so on. And as you all know, there are a wide range of sequencing platforms that are currently available, some that are more portable than others. We began with Roche 454 in 2005, and I'll show you some of that early work. It's now become much more universal. The other thing that's been very helpful has been the addition of capture, because when you can capture infectious agents, whether they be bacteria or viruses, using oligonucleotides and magnetic beads, you can be much, much more efficient. And this drops the cost of doing the work and enhances the sensitivity by something like 1,000-fold. So if you haven't started to use this for discovery, I would encourage you to do so. Most of us are now using it for characterizing SARS-CoV-2 sequences. So we've identified at this point more infectious agents than I can count. I would say probably 5% have some sort of clinical or economic relevance. And then we will find much, much more, and there's really no limit to the amount of sequence that one can find using these approaches. The other thing that I want to emphasize, very important for those of you in this field like of vaccinology, is the importance of serology and recognizing that there is now a whole series of high throughput approaches. One can use phage display, one can use microarrays. The advantage of the latter is that they can be so discrete that you can find signal in association with diseases I'll show you, with acute flaccid myelitis and some other disorders like encephalitis. So by using these approaches, you can go back and you can ask whether or not there are specific peptides that can allow you to differentiate infections with different agents. And then as you winnow these down, you can move them to less expensive platforms that will allow you to do the same sorts of things that many people now do with BioFire or consensus PCR. The first outbreak of infectious disease that was characterized using high throughput methods was Lujo, a contraction of Lusaka in Johannesburg, back really in 2008. When working with colleagues in South Africa, we found that there was an individual who was a travel agent who was working in Zambia. She became acutely ill with a hemorrhagic fever. She was airlifted from Zambia to South Africa. En route, she infected a paramedic, the nurse who cared for her at the other end, and the paramedic, somebody who cleaned the room. All four of these individuals died. The only person who survived was this 47-year-old nurse who took care of these people who became ill in the hospital. And she survived because she was given ribavirin because we identified the infectious agent. There haven't been any other similar sorts of advantages that I've been able to point to in terms of introducing antivirals, but we have had successes, you'll see, with antibacterials, and I think this is powerful. Through that work, we identified Lujo virus, which turned out to be something intermediate in sequence between the New World and the Old World arenaviruses. Fortunately, as I said, it was sensitive to ribavirin. Another example was MERS. Some of you on this call maybe were involved with this work early on. This, of course, was the famous Bisha patient. The sequences obtained of the agent at Erasmus University using cultures that came from Zaki. I was contacted and asked to help by WHO and the Kingdom of Saudi Arabia in an effort to try to identify the origin of the virus, which we did by going to Riyadh. And here I am with the son of the man who died, the index case, John Epstein from EcoHealth. This was some kid who wound up in several pictures. We still don't know who he was. And this was his brother who helped me, was very, very helpful. Now, when we began looking originally for this agent, we were focused on bats because of SARS and some of the other things that we wanted to study. And we were told that there were no bats in Saudi Arabia, which of course isn't true. We had to first prove this to the Minister of Health and the Minister of Agriculture by showing him examples of bats and bat guano and bats flying around behind this man's place of business. But at the end of the day, as we began characterizing these, it became clear to us that we were never recovering virus, and we only recovered fragments of virus that were less than 200 nucleotides in length. Now, he also had 4 pet camels who he swabbed, and that proved, of course, to be the important clue. So this was the bat from whom we isolated partial sequence, and this was in close proximity to him. But most important link came about through analyzing the camels. So based on the studies that we had done with his 4 pet camels, because we could not bring samples back to the United States because of concerns of foot and mouth disease, we assembled a mobile lab and sent it out and began analyzing camels throughout Saudi Arabia. And when we used serological assays as well as PCR, we found that amongst the adult animals, 90% of the camels, including in the empty quarter, had evidence of previous infection with MERS-CoV. Seropositivity was less common in juveniles, but when we went looking for infectious agents themselves, the reverse was true. Juveniles were much more likely to have infectious virus as well as nucleic acid than were adults. So it looked like many of the coronavirus infections that we see in humans as well. We then went looking for examples of how these people could become infected. We looked at water pipes, which are very commonly used. We didn't find any viable virus there. But when we began looking at slaughterhouses, we found that slaughterhouses that did not wash meat with high pressure hoses prior to assembly for shipment to supermarkets were much more likely to have infectious virus present. So we presume what's happened is young camels are infected. When these camels are sacrificed and sent off to supermarkets, people become infected in large cities like Jeddah or Riyadh if they're not coming in contact with camels because they come into contact with camel meat. I'm going to shift now to talk about bacteria because it's the same sort of principle, and it illustrates, again, other targets that I think vaccinologists might approach. We were asked by Soumya Swaminathan, who's now at WHO, to try to figure out why so many children were developing encephalitis in this area known as Gorapore. And what we found was that there were literally hundreds of children being admitted, so many that they ran out of space and they were building a new hospital. And the explanation came about when we flew into Gorapore, and we saw that these whole areas were flooded. And as a result of the flooding, there were rodents that were trying to escape. And these children, known as the Musahar, who would capture these rodents, would kill them because they were being paid to do this to protect wheat and rye crops, were the ones who were becoming infected. Now, many people had already looked over these sorts of materials from these children, looking at spinal fluid as well as blood, and had found a wide range of infectious agents, many of whom I think should have been resolved early on. What was most interesting to us, however, was that there were these rickettsial species, as illustrated here, Orientia tutumushi and other rickettsia that accounted for disease. And once these were identified, we then created serological assays. And by using a combination of BACCAP-seq that allows us to capture with very great sensitivity bacterial sequences that are present in spinal fluid and antibodies, or the two in concerts, we were able to resolve something like 45% of the encephalitis. And this then led to treatment of these children with tetracyclines, which completely obviated that outbreak. Here's another example of where serology became extremely important. This is the disorder known as acute flaccid myelitis, or the acronym AFM. These are children who present with a polio-like illness. It appears every other year with a peak. We didn't have it last year, presumably because we had so many people who were masking. And as we began looking at that, we didn't find any nucleic acids in spinal fluid. When we looked at blood, there wasn't anything that was particularly different. When we looked at AFM or normal adults or children with Kawasaki disease or non-AFM, but when we used the serochip that allowed us to discriminate between closely related enteroviruses, we found very clearly that there was signal in spinal fluid for EVD68 that was present only in individuals with AFM. So it's another example, I think, of how people are beginning to use serology to identify targets for public health and presumably for vaccines. The last example I'm going to show you of this sort has to do with stillbirth, which is still an extremely important problem in the US as well as the rest of the world. If you look at placentas taken from children who are stillborn, here you see examples in red, high levels of Prevotella, Peptostreptococcus, Corynebacterium, and GBS, as well as ureaplasma, versus the markers we see in normal placentas. What we found was that there was a very high frequency of infection with group B streptococci. And the question then that came about is, why would that be the case if we have these streptococcal vaccines that we used. Now, this is illustrates the importance of group B streptococcus. Less than 23 weeks and after 39 weeks gestation, women are typically screened very late in pregnancy. The trivalent vaccine is given late in pregnancy, missing 33% of the stillbirths located over here. And furthermore, the initial vaccines that were being used missed two serotypes, 2 and 5, that were responsible for 45% of stillbirths. So by not giving the vaccine in a timely fashion and not having inclusive approach to all the serotypes, we miss many of the GBS-associated stillbirths. And I can say that additional serotypes obviously have been added, but I still think the vaccine is given too late. Another important problem that we've been focusing on recently, where vaccines are also going to be critical, has been in food security. Initially became involved with this, again with 454 technology, looking at heart and skeletal muscle inflammation in salmon. And if you're eating salmon these days, the vast majority, I should tell you, is farmed. We've also done similar work with tilapia, which is extremely important in the developing world. The heart and skeletal muscle inflammation of salmon, which is illustrated here, is devastating to this industry. You can see that the fish are runted if they don't die, very high mortality. This turned out to be a novel reovirus that required very complicated bioinformatics for identification. Even more interesting was the work that was done with tilapia. Now, tilapia are farmed worldwide. We became interested because there were losses in Israel, Colombia, Ecuador, and Thailand that we were asked to help with. And intriguingly, when we did our high throughput sequencing, we didn't find anything that looked like a virus at all. All we found were these 10 segments that were intriguing because at the termini of these segments, it ranged from 500 nucleotides to 1.5 kilobases in length. There were similar sequences suggesting that they might serve as binding sites for a polymerase. When we forced it, what we found was that there was one area in the largest of these coding segments that looked somewhat like the polymerase of influenza C. And it turns out that, in fact, this is a sort of a novel orthomyxovirus, which has now been addressed as important by the FAO of the United Nations, because it also is destroying this very important protein source and economic source. You're going to be very busy and continue to be busy because the number of agents that will continue to emerge, bacterial and viral, is staggering. And that's a function of changes in land use, food industry, our susceptibility to infection, and of course, climate and weather. And I've illustrated some of the ones that I think are going to be important here. Climate change is not to be underestimated. If you look, for example, at the distribution of mosquito vectors for yellow fever alone from 2020, you can see that it's expanding further north as well as further south. So we are going to see more dengue, Zika, chikungunya, and other infectious agents. We've also begun to see a spread in the distribution of tick species. Well, whatever emerges in the developing world is rapidly transitioned to other parts of the world. So this is a New Yorker's view of the world, and you can see the number of passengers, close to 140 million, 1.4 million tons of cargo. That's into JFK, LaGuardia, and Newark. If we look at Europe in contrast, you can see that it's even more. So you have 3.35 trillion tons of cargo that's coming into Paris, Ireland, and the UK. And these are all the various airports that we've identified in those regions. We also have a global animal trade. This is not only in legal animals like pork, beef, and poultry, but also in animals that are part of the pet trade or the medicinal trade or exotic meat trades. We don't know the full extent of this, obviously, because the majority of it's illegal, but the estimates are that the US received some 90,000 kilograms, Charles de Gaulle roughly 273,000 kilograms. So we don't really know what's happening in China because we don't have any direct data. A few years ago, we looked at JFK Airport at bushmeat that was coming in. You can recognize here heads of primates. We found a wide range of retroviruses and herpes viruses and examples of multiple infections simultaneously. We've tried to estimate the zoonotic pool. Several years ago, Steve Morse suggested that there might be 1 million viruses of mammals yet to be discovered. We tried to extrapolate from work that we were doing in Bangladesh, using at that time consensus PCR, found 50 novel viruses, and estimated by extension to all the known mammal species that might become infected, that at any given time point, there might be as many as a minimum of 320,000 mammalian viruses awaiting discovery. And at that point, we projected a cost for characterization of these of about 6.3 billion US dollars, or 1.4 billion for 85% of the total diversity. Now, obviously, just finding something is not going to tell you whether or not it's capable of causing significant disease. But the argument we made was that even if 1% of them did, that the impact was not tolerable. So here's what I try to show to ministers of health around the world: the cost of AIDS, cholera, BSE, SARS, so forth. And you can demonstrate that this has been borne out in COVID. Mauricio Cardenas suggested that the global economy has contracted dramatically in 2020 with an estimate of $22 trillion in lost output. So in addition to the loss of life, what people want to see is the way you're going to make an economic case for it. Now, my work in China really began back in 2003. This is a picture taken with Chen Zhu, who was then vice president of Chinese Academy of Sciences, Thomas Breeze and myself, working with Rufer Yang, who was showing us some of the work that he was trying to do with characterization of these infectious agents. And at that time, I'm sitting with Zhongnan Chen. And then you can see us almost 20 years later. We've lost a little hair, put on a little bit of weight, but basically we're having the same kinds of conversations at this point in Guangzhou Airport. Now, many of you are going to be talking about SARS-CoV-2 and so forth. I just want to make only one point, and that with respect to vaccines. And that is that from a public health vantage point, we wish that we were putting some effort, more effort, not only into reducing and mitigating disease, but finding ways in which we can prevent transfer of infection. And I would encourage those of you who are interested in oral and mucosal vaccines or nasal vaccines to push this way. So of the vaccines in clinical development, only a handful are actually focusing on mucosal immunity. And this is something that I wish you would all emphasize. I'm going to talk briefly about long COVID, because this is something that is very much on people's minds, and we can bring some insights to this from work that we've been doing with chronic fatigue syndrome, myalgic encephalomyelitis. So the post-COVID sequelae are familiar to many of you. This is a review article that I wrote with Tony Komaroff that's readily accessible to many of you. I'm just going to illustrate a few points that are similar. So ME/CFS is something we've known about since the 1980s, but historically, there were probably cases that be documented at least back to the 1700s. So these are individuals who have profound weakness and fatigue that's not refreshed by rest or sleep. Some of these individuals have a dramatic drop in blood pressure and increase in pulse when they stand up, presumably because of misfunction of the autonomic nervous system. And you frequently have this cognitive impairment that's described by patients as brain fog. They have immunological abnormalities, including polyclonal B cell activation and metabolic disturbances. We've been looking for evidence of infectious triggers because the majority of patients will report some sort of a flu-like syndrome prior to the onset of disease in the weeks or months that are antecedent to the development of ME/CFS. There's another disorder that was described in the Gulf War where people received large numbers of vaccines, up to 14 over a period of a couple or 3 days prior to being shipped to the Gulf. And these people also present with fatigue, muscle and joint pain, headache, and so forth. So you can begin to see that there are similarities between ME/CFS, this Gulf War illness, which may reflect exposure to multiple antigens, and post-COVID. Though obviously there are some distinctive differences that probably reflect the unique aspects of SARS-CoV-2. One of the things we found with ME/CFS is that individuals have an antigen-driven clonal B-cell expansion. This is something that I think we should be looking for in long COVID. And there's another fascinating phenomenon that we've identified by culturing peripheral blood mononuclear cells immediately as they come from the patient's body, stimulating them with LPS, poly-IC, the superantigen staphylococcal enterotoxin B or Candida albicans. And what we see in this instance is that women, particularly women over the age of 45, have dramatic elevations in IL-6 in response to this superantigen stimulation. In addition, if you look at the metabolomic profiles of these individuals, what you find is that they have an abnormality in the TCA cycle such that they get a backup of citric acid. So here you can see the TCA cycle, for those of you who don't remember it. Here we're looking at what happens before they're given an exercise stress or after an exercise stress. And you can see that the levels of citric acid go up dramatically from here to here. In contrast, in the controls, they drop. So there's an adaptation which does not occur in individuals with ME/CFS. And this is just a blog to illustrate the same point. So we're beginning to understand, at least in ME/CFS, some of the cytokine abnormalities and some of the metabolomic abnormalities. And I would encourage you, those of you who are interested in the pathophysiology of long COVID, to examine these same sorts of things. Now, this portion may not be applicable, but we've also done microbiome studies and feces of these individuals, and we find that people with ME/CFS have dramatic reductions in levels of butyrate-producing bacteria. So this is the normal, this is the individual who's the case. And what we find is that these individuals are deficient in the production of butyrate. And the importance of this is that butyrate is an anti-inflammatory compound. It supports gut motility, energy source, and so forth. And it's also critical in the functioning of the central nervous system. As we look into the future, the question is what we can do to ensure that we never have another one of these. And this is really where I began back in 1981. So this is now 40 years downstream. Now, Jeff Skoll, who's the first president of eBay, has decided to invest in this. And he's created this program called the Global Alliance for Preventing Pandemics. And the focus is not so much to try to characterize agents in the developing world, but to build capacity abroad. And the idea here is that we bring people to Europe or the United States, we get them trained so that they can begin to focus on doing work in recognition of the international health regulations of the UN that started back in 2005. So we've done some work of this sort in Fiocruz with training in Zika and dengue surveillance. People are now doing much of their own work there very nicely. We're doing with Liberia. We were successful in monkeypox control. We're beginning to train them in NGS. They now have a MySeq that they're beginning to use with some success. Similar work in Mali, in Indonesia, where we've helped them build a BSL-3 laboratory. And we've begun to extend this work now to thinking about Peru, the Democratic Republic of Congo, Uganda, Kenya, the Institute for Primate Research in ILRI, and in Mongolia. And I'll try to give you a couple of examples of those now before moving on to other things. We began a convalescent plasma trial on RCT back in 2020 in New York, primarily at Columbia. But when the outbreak moved on, we had to shift to Brazil, and they were very rapid in adopting these methods and moving it forward. We've done some work in Mongolia trying to investigate case surges there, as well as similar work in Liberia. And the point I'd like to make here is that the first authors on these papers are native to Mongolia, as well as to Liberia, because what we're trying to do is to build local capacity. Here's an example of what we learned in Mongolia. Mongolia responded very early with a lockdown. They were able to keep things under control. They began vaccinations. They became a little bit more lax in terms of what they dealt with, in terms of social distancing and masking and so forth. And then they had these spikes. And the question was, did these spikes reflect the introduction of Delta or not? So the first thing we did was capture sequencing to try to identify the agents. That we found was that the vast majority were Alpha. They were not Delta. I'm sure this is no longer the case, but it then was. And we began then looking at the vaccine programs because they'd used primarily Sinopharm, to some extent Sputnik, and to a lesser extent Covishield and Pfizer BioNTech. And when we used the Euroimmune system to try to measure levels of antibodies that we found in the vaccinees, we found that levels were far lower in the individuals who'd received Sinopharm and Sputnik than they were in the individuals who'd received the others. When we then did neutralization, and our neutralization test is perhaps different than others, it's an endpoint neutralization that's based on PCR analysis of supernatant that requires absolutely no signal whatsoever in PCR. What we found was that when we were looking against the original prototypic strain that we received from Washington, Pfizer BioNTech, roughly 80% of individuals had antibodies, Covishield about 50%, Sputnik 25%, Sinopharm 20%. And not surprising, I'm sure to all of you who know far more about this than I do, when we began looking for neutralization against Delta, it was lower with Pfizer, about 30%, somewhat better with Covishield, but dropped dramatically with Sputnik and with Sinopharm. The problem with this, and as we're thinking about trying to adapt and come up with new vaccine strategies, is that the most prominent trading partner for Mongolia is China. So this is not something that they really want to publicize. So there are always political challenges as well. I'm going to close by talking with you about some of the problems that you and we face in trying to introduce diagnostics, vaccines, and discovery into this field of infectious diseases. I've been struggling with Wakefield now since that paper first appeared in Lancet, nearly 20 years ago. It took a very, very long time to disprove it. We worked with John O'Leary and Orly Shields, who have been people who've been originally involved with the original paper by Andy Wakefield. Nonetheless, people are still holding and supporting the work that he proposed, even though it's been largely disproven. And we have huge problems with vaccine resistance in the United States and elsewhere. And we'll talk about misinformation because that's critical. So we have problems with not only the resistance to vaccine, but the use of a wide range of anti-infectives that don't work. The most recent, of course, is ivermectin. Now, when I was sick, I was treated with hydroxychloroquine because at the time, that was the drug that we were all pushing. Now, this is evidence of how Andy Wakefield is still very active. So he put in a film called Vax that went into the Tribeca Film Festival. I was able to keep that out. Now we have Plandemic, and Plandemic is attacking not only me, but Tony Fauci and others, probably people who are on the call. There are some people who should know better, who have been promoting this. RFK Jr., for example, Instagram, Facebook, and others that have become very, very common sources of information. Now, in the movie Contagion, we talked about some of this with this character, Alan Crumwitty, who was pushing a drug called forsythia. And here we have the hydroxychloroquine, and we have a variety of other things that have been pushed by Donald Trump and others. We tried to deal with this early during the course of the pandemic. So this was a PSA released by the cast of Contagion in March of 2020. And you can see
everything you're going to hear from us has been vetted by public health experts and scientists.
A pandemic means that the virus is everywhere, but it won't be every place at the same time.
We all want a cure, but the health of our society is in your hands.
It needs us to survive. Let's not give it any help.
now when we went back to the cast and said, we should really do something to promote vaccines. They said to us, nobody's going to trust us because we're celebrities. We need to show examples of people who look like individuals who would run into during everyday life. So that's precisely what we did.
COVID-19 vaccine out of fear and out of hope. I'm taking the COVID-19 vaccine because I was fortunate enough to survive it.
Trust me, you don't want what I had.
No one should have to go through such suffering. I beat cancer twice, and I am a COVID survivor. Our community has been devastated.
So by using these sorts of media approaches, we can reach people we wouldn't otherwise reach. In March of 1999, I went to a US Department of Defense meeting that was held at Fort Monroe, Virginia. There were 3 virologists, 1 animal virologist, me, a plant virologist, and a computer virologist. And I hadn't really thought about computer virology, but it's become extremely important during the course of the pandemic. This whole field of of things going viral really goes back to advertising in the late 1980s with the discussion of some way in which you could promote ideas. It has now taken off in a way that none of us could have anticipated. So if you look at the number of emails that are exchanged every minute, it's close to 200 million. Mobile texts, GIFs, videos, Facebook logins, people tweeting, and so forth. And this is extremely important because these social media platforms drive the information that people exchange and are probably the largest single impediment that I can think to promoting science. So while I try to focus to some extent on doing whatever I can in writing scientific articles and such, I also try to do things in foreign affairs, Wall Street Journal, Bulletin of Atomic Scientists, going on Fox News, talking to the Daily Mail as well as the BBC. And I would encourage all of you who are scientists to make this effort because this is the only way in which we're going to shut down misinformation. These are some of the movies in which I've been involved. One of them is the new, excuse me, I'm sorry. I guess this film is finally going to come out. Plague Inc., which was a game. This, which is about HIV, Utopia. And I was very gratified a few months ago to see that Matt Hancock liked the Matt Damon film, though it isn't a Matt Damon film. It was really written by Scott Burns and done by Steven Soderbergh. And I don't know how much time I have. Do I have 3 minutes? Yay or nay? Someone,
Yes,
anyway.
you do. Yes, you do.
Can I have 3 minutes?
Absolutely.
Okay, so let me show you some highlights of Contagion.
Well, in 1918, 1% of the population died from Spanish flu. It was novel like this. No one had ever seen it.
1% of America.
1% of the world. With the new mutation, we are predicting an R0 of no less than 4. Without a vaccine, we can anticipate that approximately 1 in 12 people on the planet will contract the disease.
So
So at this point, I think we have to believe this is respiratory, maybe fomites too.
What's that, fomites?
It refers to transmission from surfaces. The average person touches their face 2,000 or 3,000 times a day,
2,000 or 3,000 times a day.
3 to 5 times every waking minute. In between, we're touching doorknobs, water fountains, elevator buttons, and each other. Those things become fomites.
Honey? Hey, honey. Yeah, Jesus. Sweetheart.
You had a seizure this morning, Beth. Have you had something like that
She have a
before?
history of seizures?
No.
Allergies, other
That,
medical causes?
I think she's
She
allergic to
fall recently?
penicillin.
Did her head in the
No.
shower?
She came back from a trip and she
What about
was-
drugs, MDMA, ecstasy?
No, we don't do that.
Okay, let's get some help.
Back.
Let's get a line here.
Centers for Disease Control and Prevention in the United States and the World Health Organization in Switzerland confirmed today that Dr. Ian Sussman of San Francisco has succeeded in growing the MEV1 virus in a laboratory setting. Officials at the CDC caution that the breakthrough is only the first step toward developing a vaccine, which is likely still months away from human trials. The WHO estimates the number of people infected worldwide to be over 8 million.
Only be able to give out 50 doses today. That's hardcore, Cynthia. I love it. But excuse me, there's a lot here.
We may never know where this disease came from, but we do know that this vaccine is a result of the courage and perseverance of a remarkable few. We shall now begin the drawing. John. First MEV1 vaccination are those people born on March 10th. March 10th.
So in closing, and then I'm happy to take questions if there are some. We're very focused on trying to create a global immune system. I think that's the point of GAPP. The other thing I didn't get a chance to talk about is the fact that I think we need the equivalent of a global FDA so that we can more rapidly develop and validate diagnostic drugs and vaccines. We've emphasized the emergence of zoonotic and high threat pathogens, the wild animal trade and the pet trade. I've not talked about international standards for research and biocontainment, but these are critical as well. Not so much because I think that there's risk that there's going to be something that's going to emerge from biocontainment, but we need to remove this criticism from the social networks. And the last point, which I would encourage all of you to join me in doing, is to promote public health education. by investing your time in entertainment, commercial, and social media to mitigate the impact of mis and disinformation. Thank you very much. And have a great conference. Manon?
Lipkin, that was really fantastic. It almost, you went with lightning speech to I don't know how many organisms. And, you know, one burning question that comes to my mind is, what could we or would you have done differently to deal with COVID early on, if anything?
So this is really a source of self-reflection and discomfort. When I was in China, everyone was masked. When I came back off the plane, no one was masked. And I struggled for a long time with whether or not we should be talking about this because there weren't enough masks at that point. And in retrospect, I wish I'd pushed much, much harder on that front.
On masks.
When I was in China meeting with Chen Zhu in Beijing, he was buying up personal protective equipment and he said, I don't understand why the whole world isn't doing this. That's all we had at the time. And we should have been much more vigorous on that point. The other issue is that we failed to recognize that the virus was already global. So if you're going to try to shut down people coming in from elsewhere and you don't have a good diagnostic test, you can't say we're just going to shut off people coming into Newark from China. I was on the last flight from China into Newark, which I believe was on the 4th, if I'm not mistaken, of February. But people were coming in from Europe, and there was disease in Europe at the time.
Right.
The other thing is, since 2003, we've been talking about these wild animal markets, and we were assured that they were shut down. And I had conversations like this in China the last week of January. They said, no, they've all been shut down. And then more recently, we've seen pictures, 40,000 animals in Wuhan that were brought in either by order or consignment and so forth. There can be zero tolerance for this internationally, not just in China or Malaysia or Africa, but in France and in New York. We have a large wild animal trade in New York. And as I showed you, these animals that came in through JFK, in which we found infections. So there's so many things that we should do of that sort. But the other thing that I am, was another issue was we had good diagnostic tests that were developed in Europe that could have been used in the United States. And instead, we went through this period where we didn't have good testing, as you know, because there was a problem with the negative control, which basically thwarted our ability to test.
Important time, yeah.
But since then, I mean, I will say this, I was very involved early on with Amazon in helping them in Italy with the Democratic National Convention and the whole entertainment industry, where they've been able to maintain content because they had very, very rigorous methods for control. And if you've seen some of those protocols, if anybody would like to see them, I'll be happy to share them, where we talked about everything from testing to isolation to vaccines and so forth. Now, at present, I think we're going to see some slippage, particularly in the live entertainment industry, about which I am somewhat concerned. But there was a point at which they wanted to open up Madison Square Garden. That's a year and a half ago. That would have been devastating. Anyway, so there are a number of things I regret.
Okay, thank you. There was one question from the audience, from somebody out of the audience who wanted you to go a little bit in further detail on the strength of a possible link, whether there's truly evidence between CFS and disease in the troops, the Gulf War syndrome that you discussed. So
we don't know so first of all, ME/CFS is a very complicated disorder. There are people who have, I would say 30% of them have a history of some sort of prodrome where they have fever, lymphadenopathy, low-grade night sweat, something suggestive of an infectious trigger. And what we think probably happens is that there is some sort of epigenetic modification that we don't yet understand in the immune system that makes people more sensitive to common pathogens, be they candida antigens or super antigens or other things that we don't yet understand. So, but there hasn't been the same kind of investment in this field that you had elsewhere. So, you know, we run one of the largest centers in the world for this. And if I were to tell you the size of the budget, you know, it's paltry.
It's too small.
Very,
So,
very.
you know, unfortunately, our hour is over, and I know that Ted Gibson and ISV is ready to release the next talks, but I think this was an amazing talk. There have been many, many thank yous in the chat. I hope you've seen them. And I very much enjoyed this. I want to thank you very much for being with us today.
My pleasure, and thank you so much. And I just want to emphasize in closing that you people are the solution. We try to identify the problems, but you need to fix it. So Godspeed.
Vaccines.
Okay.
Thank
Bye-bye.
you.
Yeah.
All right. Bye-bye.